Hello everyone, in this video we're going to start a new lesson which is lesson 6, which is about DNA structure and function. We want to talk about more about the DNA. What is the function of the DNA? How we can make from the DNA the protein. Okay, let's start first here. What is the DNA? So the DNA, which is the genetic material in cell, is contained in a molecule called it actually contained a molecule which is deoxyribonucleic acid, or we can say which is a DNA. Scientists describe DNA as containing a code. Actually, the DNA in our cells, it's like a code. Yani you can imagine the DNA, it's like if you are reading from, it's like a book and it has code to do something. Yani it just gives the information. It doesn't do anything. Just give the information. Give the information for growing, for dividing, for making a specific protein. Okay, so this DNA, it's like a code. So a code, what is a code? Is a set of rules and symbols used to carry information. Okay, we want to study exactly what is the structure of the DNA. It contains what? You remember we said it's a nucleic acid before, and we said the nucleic acid, it's made of a nucleotide. We will talk about all of these points. We will understand exactly what is the structure of the DNA. So to understand how DNA function, you first need to learn about the structure of the DNA, to know how it works and how it starts to give the information to make a specific protein. Why we are talking about a specific protein? Because actually the DNA, it's giving what? The information. Most of the functions in our cells, they are made by a protein. That's why protein is very important. So again, let's talk about this one here. For the protein, most of the function are the things that the DNA doesn't do anything, doesn't do the growing, doesn't do the dividing. A protein do most of the work of a cell and also make up much of the structure of a cell. Okay, how the scientists, before we start to talk about the structure, how the scientists found the structure of the DNA? What did they do? Actually, is it by overnight they found what is the structure of the DNA? No, actually many scientists from all over the world contributed to our understanding of DNA. Okay, some scientists discovered the chemicals that make up DNA, and others, they learned how these chemicals fit together. Still others determined the three-dimensional structure of DNA molecule. Okay, so experiment and imaging techniques have helped scientists to infer the shape of the DNA. You remember when we talked about the DNA and the meiosis and mitosis, when we talked about the chromatin and the chromatid and the chromosomes, I told you in the most life of the cell, it's like a chromatin. They are thread-like structure and they look like a spaghetti. Actually, the DNA, it's very long. It's very long structure. It's not very small, but it's very compact to be fit in the nucleus. Listen to this.
DNA is usually coiled up tightly within your cells. If all the DNA in your body were stretched out in a line, it would reach from the Sun to Jupiter and back again more than 8 times.
Can you imagine?
There's so much DNA in your body because it's got a lot to do.
Yeah, can you imagine how the long of the DNA? You're talking about all the DNA in your body. Actually, one DNA, it's like equal to like 2 meters. Yani, can you imagine? And all of them together, they will make like go to the distance from the Sun to Jupiter, and it like go and back again like 8 times. Can you imagine this?
DNA stands for deoxyribonucleic acid. DNA is the genetic material in cells that controls cell functions and determines many of our traits. Scientists have been unraveling the mystery of DNA for a long time. Click the dates on the timeline to learn more.
Okay, let's start to see how the scientists contributed to discover the structure of the DNA and how it works. Let's start with the first one. In 1857.
Gregor Mendel was an Austrian monk who performed breeding experiments with thousands of pea plants. He observed that the offspring of pea plants had some of the same traits as the parent plants. Mendel hypothesized that some factor must be passed down from parents to their offspring.
So Mendel said there is a factor, but he didn't know what is that factor. Actually, at the beginning, they thought it's a protein. Actually, for this one, let's continue.
Swiss biologist Johann Friedrich Miescher isolated a substance he called nuclein from human white blood cells. We now call that substance DNA. Actually,
for this scientist in 196, a substance that will he found this substance, but he didn't know what is it exactly, but he didn't know this one is DNA. Let's continue.
Phoebus Levine published a paper that describes the components of DNA. DNA is made up of a sugar, a phosphate, and 4 types of nitrogen-containing bases.
Okay, so for Phoebus Levine, what did he say? The DNA, he published a paper that this paper, a paper on a nucleic acid. His research helped scientists to determine the DNA is made up of sugar. Okay, the first one, the DNA is made up of sugar, phosphate group, and 4 types of nitrogen bases. What are these 4 types of nitrogen bases? Adenine, thymine, guanine, and cytosine. And he found this phosphate group P as sugar, and each sugar is linked with what? A nitrogen base. A represent adenine, T represent thymine, G represent guanine, C represent cytosine. Okay, let's continue.
Adenine, thymine, guanine, and cytosine. Bases are often referred to by their first letter, A, T, G, or C. Each base has a different shape.
Look how they have adenine A, how it looks. Look at T, thymine, how it looks, and they have different shapes. And G, guanine, C, cytosine. Each one of them, it has a different shape, as you see. So in this model, if I want to talk about this one, if I want to ask you about this one, the first one here. Okay, I'll put a circle here. For this one, what do P represent? Maybe I'll ask you, what is P represent? P represent the phosphate group. Then, what about S represent what? Represent sugar. What about A? Represent a nitrogen base, which is adenine. Okay, adenine base. Maybe I'll give you this one here. P represent phosphate, S sugar, C cytosine, and so on. Okay. Let's continue. How the scientist first Mendel, he said that they are factored. Then Friedrich Maysa, he said there is a DNA, there is a substance which is called from the white blood cells. Then Levine, he published a paper about the component of this DNA, and it contains sugar, phosphate, and the nitrogen bases. Listen now, what did the scientists discover then?
British geneticist Frederick Griffith showed that an unknown substance within dead pneumonia-causing bacteria could be passed on to formerly harmless bacteria, so that the formerly harmless bacteria could cause disease in rats. The unknown substance was later found to be DNA.
No, till that time they still saying this is unknown. Then they found this factor can inherit like traits to for the other organisms or makes disease. Like this one here, which is 1928. Let's say about. He just say like this about it. We didn't mention anything. Look at this one.
Oswald Avery, Colin MacLeod, and Macklin McCarty repeated Griffith's experiment with some modifications. Through their new experiment, they were able to show that DNA was the substance that controlled heredity in bacteria.
Okay, let's look at this one now.
Biochemist Erwin Chargaff discovered that the amount of DNA varies from species to species. He also discovered that within a species, the amount of adenine within DNA is always the same as the amount of thymine. In addition, the amount of guanine within DNA is always the same as the amount of cytosine. His findings are known as Chargaff's rules.
Chargaff's rule is very important to know that, very, very important. The amount of adenine equal to the amount of thymine. Amount of guanine equal to the amount of cytosine, and always adenine linked with the thymine, guanine always linked with the cytosine. Very, very important, this one.
Rosalind Franklin and Maurice Wilkins produced images of DNA using X-rays. These images provided valuable clues about the structure of DNA.
Here they just used the X-ray to study the structure of the DNA. They found it like this. Okay, let's continue. What did the scientists do?
Alfred Hershey and Martha Chase conducted experiments with viruses to confirm that DNA, not proteins, carry genetic information.
So here they discovered from the virus, they knew that the DNA, not protein, they are carrying the genetic information, because they found at the beginning the protein is carrying the genetic information. Okay.
James Watson and Francis Crick used the images produced by Franklin and Wilkins to construct a model of DNA. Their model showed that DNA was a double helix, which is a twisted ladder-like structure. Their model followed Chargaff's rules.
Now we will talk about the structure, how a double helix twisted structure. We will talk about it. I want you to understand for each scientist how each one of them contributed to know the structure of the DNA. If you have any question, you can, next time we will continue about the structure of the DNA. If you have any question, you can send me your questions. Thank you for listening.